Hello everyone. So in this video, I will show you different examples of how we can use our Rothia model to address different questions related to bacterial metabolism. So here, first of all, we start by opening our Jupyter notebook via the command line using the command Jupyter notebook, which directly takes us to the browser of our preference. And here we can open our notebook with all the functions that we will need. So for this tutorial, we will need 4 packages and their associated libraries, so that we're gonna use the Python language and the 4 packages Cobra, Pandas, NumPy, and Matplotlib for plotting figures. The next step here is to set some parameters that we will need later for the diagram. And here I'm just defining all the transition methods to have some like a global list to store them for later use. So the very first step here is to import and read the model, the SBML model, using the Cobra function read SBML model. And as you can see here, we get the number of metabolites, reactions, groups, the objective function, which is the biomass reaction, and also the cellular compartment. So first I would like to take you through a typical test that I always do with a model to check on which growth media they can stimulate growth, positive or no growth at all. And this is to check different growth media in different environments. So here we define a global function that takes a model and defines an exchange reactions and sets the medium of preference within the metabolic model. Next, we import the table with a media of interest. We can use the Pandas data frame to store the table for easier use. And here the example is for the TSB medium. Now we're going to use the Cobra package, and firstly we set the medium using the function that we already defined. And then we define the objective function, which is here the growth, the cellular growth. Here we optimize the model and we calculate or we store the growth rate. I also calculate here the doubling time in case we need it for later use or validations or comparisons. And the same we can do also anaerobically, turning off the uptake of oxygen, set the objective function, optimize, and print out the growth rate and the doubling time. And using here the with functionality of Python. So with model, all changes done within this clause are done reversible. So at the end here, we'll get again the initial conditions within the model and settings. Second example here is with the synthetic cystic fibrosis medium. Again, we read and import a table with a stored medium of interest, and we select the columns related to the SCFM medium. In this case, as you can see, our medium has 37 metabolites. And again, using with model, we set the medium of interest here. And as we explain in detail in our manuscript, there were some implementations needed in order to simulate positive growth with this medium. So here I'm also defined the implementations, and I set the objective functions to be the growth, optimize the model, and print out again growth rate and doubling time. And I repeat the same procedure for anaerobic conditions. Now let's see how, if we have a predefined medium, what else we can do or which individual modifications we can do. So for example, if we have the SCFM medium and we would like to see what is the effect of turning off glucose uptake on the metabolism is, we can do this by going and turning off individually the glucose uptake here. And now, as you see, we have zero growth. So glucose is in principle essential for growth in the SCFM medium. And with this, we can move on and see which metabolite or which uptake rate, if we enable it, we can get a positive growth, even if we have a glucose turned off. And this is what we do in this part here. And the result here is a list of different uptake rates, which could lead in a positive growth even if we turn off the glucose. So the next step is a simple quick check in which we examine the different oxygen uptake rates or oxygen levels in different environmental conditions. And in this case, I will show you two different media, the TSB and the LB medium, and how the different oxygen levels influence the cellular growth. And for this purpose, we define here a function called check oxygen effect, in which we give any medium that we'd like, and the function goes on and defines the medium and sets a different oxygen rates starting from 0 up to 1000. Finally optimizes the model and gives out the computed or predicted growth rates. And so if we do this for the LB and TSB media, we can collect all the results within a data frame to have a better overview and be able to analyze them quicker. And we can, of course, plot nice line plots showing the different oxygen levels for the different medium here. So on the left, we have the TSB, and on the right, we have the LB, and we have all the computed growth rates starting from 0 up to 1000. So in this example, we can see what we can get, like if any compound of interest is modified or the rate is modified, like the export or the uptake rate, and how this affects the bacterial growth. And this is just an example with oxygen in different media, but we can do this for any compound defined within the model, and that is of high interest to us. And we can also manipulate different metabolites or sugars or different carbon sources and so on. So finally, let's move on to the gene knockout test, for which we will need the LB medium and the M9 medium. And so we would like to test again both aerobic and anaerobic conditions, and we will use the COBRA function called single gene deletion. And here we collect again our results into a data frame. So we have here over 370 genes, and we have 4 different columns for the 2 media and the 2 conditions respectively. And here we get the growth rate after turning off each gene in each condition. And we can do this also for pair of genes using again a predefined COBRA function called double gene deletion. And this time here our table is way more bigger, is over 69,000 rows. And this is because the algorithm tests every single pair of genes for the medium of interest. In this case, I chose LB. And finally, we can manipulate or we can conduct gene deletions manually, which means we can take any 2 genes of interest and knock them out individually and then optimize the model, selecting the correct objective function, store the objective value, and see the effect of this double gene knockout into cellular metabolism. So I hope this video helped you to get an overview of how we can use a Rothia model for any kinds of metabolism related questions. These are some basic functionalities and some basic predictions that we can get, but there are still many more options and tasks that such a model can do for us. Thank you very much, and for any questions, don't hesitate to contact us.